good evening. This is Kin Jimenez from the Entrep 1A, and I'm going to vlog about the gene therapy. The first question is, when was the first gene therapy products were approved? Approved in 2017 in the US, with more likely to be approved in this year and thousands more clinical trials, including a trial for Rett syndrome, sponsored by Avexis, a Novartis company, expected to get underway this year. So what exactly is gene therapy? It's the addition of new genes to a person's cell to replace faulty genes in an attempt to prevent, improve, or cure diseased genes. Needs a bit of help to get into cells. One way they get that help is through a virus. Now think about that virus is normally do. They're really good in getting into our bodies and getting us sick. They do that by injecting their genetic materials into our bodies. So scientists had a great idea of hijacking those viruses to deliver genes into our cells. But don't worry, they removed the parts that makes us sick. Second question, what are the basics of gene therapy? Well, genes are made of DNA, which are the blueprints, which are built up to the enzymes or proteins that do the work for your body. As far as we know, humans has 20,000 to 25,000 genes, and most people has 2 copies of each gene, inherited from each of their parents. They are the building blocks of our physical makeup, from the color of our hair to our athletic abilities and our immune system. But sometimes genes aren't built correctly, and a small adjustment to the DNA in our genes can change our proteins' work, which can affect on how we breathe, talk, or digest food properly. If genes are blueprint to our body, gene therapy can fill in some missing drawings. Gene therapy is the introduction to remove or change in genetic material, specifically DNA, or its molecule cousin, RNA, into the cells of the patient, to repair or change a gene-specific disease. Transfer genetic material then changes how a protein or a group of protein is produced by the cell. It can reduce levels or certain disease-causing proteins, increase production of disease-fighting proteins, or even producing new or modified proteins within the cell. So, you may be curious on how a gene and a genetic material it holds is transferred to a cell during the gene therapy. Well, a gene cannot be directly inserted into the cell. Instead, a carrier, also known as vector, helps deliver the gene. In vivo therapy, the genes introduced directly into the cells, while the cells are still in the patient's body. In ex vivo gene therapy, cells are removed from the patient's body. The genes are added to those cells in the lab, and then the cells returned in the patient's body. The right approach depends on the disease that the patient has and the tissue that's affected, and that would tell us what's the best way to approach this. How do vectors do? It is a vehicle designed to deliver a therapeutic genetic material, such as a working gene, directly into a cell. Typically, vectors are derived from viruses because of the viral genes are removed, and the vectors are modified to only deliver therapeutic genes. This viral vector uses the shell or capsid of the virus to transport the working genes into the target cell. Think of them as envelopes used to deliver a specific message. Researchers only use vectors that are extendedly studied, determined to be safe to use in humans, and have the ability to target the specific cells. How many types of viral vectors do you know? There are 4 main types of viral vectors, each with their own unique characteristics and limitations. First, let's talk about adeno-associated viral vector. AAVs are typically used to deliver the smaller DNA's package or genes. So the size capacity of this vector may determine which disease they may target. They're safe and efficient for in vivo gene therapy approaches, in which the therapy is injected into the body, delivering new instructions directly into the cell. So, if it's taken up by a cell that divides, the therapeutic gene won't be copied with each cell division. It may be lost over time, thereby diluting the treatment effect, such as in the liver nervous system, such as cells in the liver, nervous system, eyes, and skeletal muscles. The advantage in using these vectors is once delivered DNA can persist in the patient's body in a long time, possibly even in a lifetime. One limitation is that many people may have prior exposure AAVs through natural infections, giving them immunity. Antiviral and retroviral vectors. These vectors can carry large genetic packages of RNA, which is then converted to DNA. During this process, the vectors integrate into the genome target cell. AAV's vectors makes lentiviral vectors and retroviral vectors best suited for ex vivo in dividing cells. T cells, which are the type of immune cells, and stem cells. Their special cells can develop many cell types. Example, which are special cells can develop cell types. Ex vivo treatment involves moving cells from the body, modifying them using a gene therapy, and then returning them into the body. There, treated cells will begin to divide and generate new cells. New genetic material is then copied into all those cells. Gene therapy using viral vectors is already treating rare inherited disease and certain forms of cancer, with many other potential uses being studied, including clinical trials.